Hello friends, today in this video I am going to discuss about the MCQs on immunology, and this is the 10th part of the series of more than 1800 MCQs on immunology, and this video will be beneficial for many life science exams like CSIR NET, ICMR NET, GET Life Science or any other MSc entrance exams, and also for medical laboratory technology exam, nurse exam, hospital management exams, FSSAI exams and many other exams. So let's start the MCQ sessions with detailed explanation on immunology. So the first question is, Myasthenia gravis is a T cell deficiency disease, a B cell deficiency disease, or a combined T and B cell deficiency disease, or an autoimmune disease? And the right answer is option D, that is myasthenia gravis is a chronic autoimmune disorder. It is an autoimmune disorder in which antibodies destroy the communication between nerve and muscle, resulting in weakness of the skeletal muscle. Here you can see this is the nerve cell and this is the muscle. So here in the junction between the nerve cell and muscle, due to this myasthenia gravis, the junction is weak, okay, due to this autoimmune disease. And myasthenia gravis affect the voluntary muscles of the body, especially those that control the eyes, mouth, throat and limbs. And this myasthenia gravis is not inherited and it is not contagious. It generally develops later in life when antibodies in the body attack normal receptor on muscle. This blocks a chemical needed to stimulate muscle contraction. Okay, so this is all about myasthenia gravis. So move on to the next question, that is, and also here you can see this is the consequence of the myasthenia gravis in eye. Okay, so move on to the next question, that is passive immunity. So passive immunity is induced by contact with allergen, or has immunological memory, or give long lasting protection, or is effective immediately. And the right answer about the passive immunity is option D, that is effective immediately. So at first we have to know passive immunity. So passive immunity is a part of acquired immunity. So what is acquired immunity? Acquired immunity where immunity that developed during your lifetime. This immunity developed during your lifetime. So not so here this acquired immunity is two types, that is active immunity or passive immunity. So active acquired immunity means, and also active acquired immunity is two types, natural or artificial. Natural active immunity means that is antibodies develop in response to an infection in your body. The immunity raised or immune cells raised against a infection. So that is a natural active immunity. And and artificial active immunity means antibodies develop in response to a vaccination. That is a active artificial active immunity. And the another one, that is passive immunity. It is also divided into two groups, that is natural passive immunity and artificial passive immunity. Natural passive immunity means antibodies received from mother through breast milk, that is a natural passive immunity. And artificial passive immunity means antibodies received from a medicine, from a gamma globulin injection or infusion. So passive immunity we have to get from other body, okay, into our cell. Okay, so this is the passive immunity. So it can be natural or it can be artificial. Natural means antibodies received from mother through breast milk from outside. So that is the natural immunity. Okay, so this is the passive immunity. So move on to the next question, that is, the following is an anaphylotoxin: C2B or C3A or C4B or C567. So these all are complement protein, okay, complement components. So which one is responsible for anaphylotoxin? And the right one is option B, that is C3A. So anaphylotoxins or complement peptides are fragments that are C3A, C4A, C5A that are produced as part of the activation of the complement system, okay. And anaphylotoxin are able to trigger degranulation, that means release of substances of endothelial cells or mast cell or phagocytes which produce a local inflammatory response. So anaphylotoxins produce a local inflammatory response, okay, due to this complement proteins. Okay, so move on to the next, that is Kupffer cells present in the blood, lymph nodes, brain or liver, and the right one is option D, that is liver. So Kupffer cell are present in liver. Here we can see this is the Kupffer cell inside the liver cell, okay, and the primary function of the Kupffer cell is to remove foreign debris and particles that have come from the portal system when passing through the liver. It is possible for the Kupffer cell to take in large particles by phagocytosis and smaller particles via pinocytosis, and these Kupffer cells are integral in the innate response of the immune system. Okay, so the next question is gamma interferon is produced by macrophages, B lymphocyte, T lymphocyte or plasma cells, and the right answer about the gamma interferon is option C, that is T lymphocyte. So this gamma interferon is produced by T lymphocyte, and this interferon gamma or type 2 interferon, this gamma interferon is a cytokine that is critical for innate and adaptive immunity against viral and some bacterial and protozoan infections, mostly for viral infection. And this interferon gamma is an important activator of macrophages and inducer of major histocompatibility complex 2 molecule expression. Okay, and aberrant interferon gamma expression is associated with a number of autoinflammatory and autoimmune diseases. Okay, so the next question is interleukin 4 is a T cell growth factor or B cell growth factor or macrophage chemotactic factor or migration inhibition factor, and the right answer about the interleukin 4 is option B, that is it is a B cell growth factor, and interleukin 4 was originally described as a B cell growth factor and now known to provide potent anti-tumor activity against various tumors including breast cancer, and this interleukin 4 can induce apoptosis in cultured breast cancer cells. So this interleukin 4 is a pleiotropic cytokine produced in response to receptor activation by Th2 type, that means the T helper cell type 2, CD4 plus T cells, basophils and mast cell. Okay, so they are activated by these cells. This interleukin 4 is activated by these T cells, basophils and mast cell. So move on to the next question, that is the clinical manifestations of cell mediated immunity include all of the following except serum sickness, contact dermatitis, autoimmune disease and graft rejection, and the right one is option A, that is serum sickness. So all other are due to cell mediated immunity but not serum sickness. So what is cell mediated immunity? Cell mediated immunity is an immune response that does not involve antibodies but rather involves the activation of macrophages and natural killer cells, the production of antigen specific cytotoxic T lymphocytes and the release of various cytokines in response to an antigen. So this is the cell mediated immunity. Here no no antibody involves. Okay, and what is serum sickness? Serum sickness is an immune complex mediated hypersensitivity reaction that classically presents with fever, rash and polyarthritis. So here you can see this is the consequence of serum sickness, and the serum sickness in humans is a reaction to proteins in antiserum derived from a non-human animal source occurring 5 to 10 days after exposure. Okay, so move on to the next question, that is major compound of complement is C3, C4, C5, C9, and the right one is option A, that is C3. So here we know that complement pathways are three types, that is classical pathway, mannose binding lectin pathway and alternative pathways. So in all these three pathway one thing is common, that is all they activate the C3 protein, that is C3 convertase. They activate the C3 protein, and ultimately then the other complement proteins are activated from this C3 protein. So C3 is the major component of the complement pathways. Okay. So the next question is, the antigenicity of a substance depends on foreignness, chemical nature, molecular weight, or all of the above, and the right answer is option D, that is all of the above are. So antigenicity is depends on all these points. And what is antigenicity? And it is depends, and also antigenicity or immunogenicity is the capability of immunogen to activate immune response. Okay. And the foreignness means immune system can discriminate foreign substances. Foreign substance only stimulate immune response. Okay. And how the size depends on it. So it essentially has a molecular weight. All antigens should have molecular weight at least 10,000 Dalton. Okay. And this antigen has a specific chemical nature, should have a specific chemical nature, mainly proteins or polypeptides or carbohydrates, steroids are and lipids are poor immunogens or antigens. Okay. So and foreignness means the the antigen should be a foreign particle to be a antigen. Okay. So move on to the next question, that is, the antibody formed in primary immune response is IgA, IgM, IgG or IgE, and the right one is option B, that is IgM. So the antibody formed in the primary immune response, that is the IgM. So IgM came first after any exposure of the antigen to your body. Okay. So move on to the next question, that is, all are lymphokines except macrophage inhibition factor, B cell growth factor, rheumatoid factor or blastogenic factor, and the right answer about the lymphokine, that is C. So rheumatoid factor. So all these three, that is option A, B and D is about the lymphokines. So except the rheumatoid factor, it is not the lymphokine. Okay. So what is the role of lymphokines? So regulatory function of the lymphokines are, they stimulate, or the function is stimulation of growth and proliferation of cytotoxic and suppressor T cells by interleukin 2, and stimulation of B cell growth and differentiation to form plasma cells and antibodies by interleukin 4, 5 and 6, and activation of macrophage system and feedback stimulatory effect on helper T cells themselves, that is by interleukin 2. Okay. So the next question is, the following is a hemagglutination test. The options are Wasserman, Paul Burnell, Well Felix or Widal test. So which one is the hemagglutination test? And the right answer is option B, that is Paul Burnell test is a hemagglutination test. And the Paul Burnell test is used to screen for infectious mononucleosis, which is seen most commonly in adolescent and young adults. And this infectious mononucleosis is caused by the Epstein-Barr virus. This infects B lymphocyte, which subsequently produce a heterophilic antibody, which is identified through its reaction with the Paul Burnell antigen and is detected by a latex agglutination method. Okay. So the next question is, the following is or are peripheral lymphoid organ or organs. So the options are gut associated lymphoid tissue, that is GALT, or thymus or spleen or all of the above, and the right answer about the peripheral lymphoid organ is option C, that is spleen. So spleen is a peripheral lymphoid organ or secondary lymphoid organ. So here spleen is the peripheral lymphoid organ or lymph nodes also. So what is the role of peripheral lymphoid organ? So the role is to trap antigens. They are the sites for initiation of most immune response and provide signals for recirculation of lymphocytes and antigens delivery to regions of increased traffic. Okay. So these are the role of peripheral lymphoid organs. So the next question is, in antigen antibody reaction, Poisson phenomenon occur when antigen is in excess, antibody is in excess, or antigen and antibody are in equal proportion, or none of the above. And the right answer is option B, that is antibody is in excess. So when antibody is in excess in antigen antibody reaction, that is called the Poisson phenomenon. So Poisson phenomenon is a false negative response resulting from the high antibody titer which interfere with the formation of antigen antibody lattice necessary to visualize a positive flocculation test. So here you can see, due to the excess quantity of the antibody, here the there is no lattice structure formed, so that's why a false negative, we will get a false negative result. Okay, so move on to the next question, that is, passive agglutination test detects specific antigen antibody or antigen antibody complex or complement, and the right answer is option B, that is antibody. So passive agglutination test detects specific antibody. So what is passive agglutination test? That is agglutination. So it is also a agglutination test done with a soluble antigen coated onto a particle. So here is the carrier molecule. So we have to add this soluble antigen to this carrier molecule, and then we add the antiserum. So here the indirect or passive agglutination done. Okay, so agglutination test done with a soluble antigen coated with a particle. Okay, so this is a passive agglutination. So move on to the next question, that is, complement is activated by antigen antibody, antigen antibody complex, or erythrocyte antibody complex, and the right answer about the complement is option D. So complement is activated by erythrocyte antibody complex. Okay, so next is, allograft is taken from genetically identical twins, genetically unrelated member of same species, or different species or self. So the allograft is taken from B, that is genetically unrelated member of the same species. Okay, so allotransplant or allograft is the transplantation of cells, tissues, or organs to a recipient from a genetically non-identical donor of the same species, and the transplant is called an allograft, and allogenic transplant or homograft. So most human tissue and organ transplant are allografts. Okay, so next is, autograft is taken from genetically identical twins or genetically unrelated member of the same species or different species or self. So autograft is taken from option D, that is self. So auto transplantation or autograft from one part of the body to another in the same person. Okay, so that is the autograft. So next one is, isograph is taken from genetically identical twins or genetically unrelated members of the same species or different species or self, and the right answer about the isograph is option A, that is genetically identical twins. So isograph is taken from genetically identical twins. So next is, Genograph is taken from genetically identical twins or genetically unrelated member of the same species or different species or self, and the right answer about the Genograph is option C, that is different species. So Genograph is taken from different species. Okay, so move on to the next, that is, Wassermann test is a passive agglutination test, hemagglutination test, or coagulation test, or complement fixation test, and the right one is option D, that is complement fixation test. So Wassermann test is a complement fixation test. So Wassermann test or Wassermann reaction is an antibody test for syphilis named after the bacteriologist August Paul von Wassermann based on complement fixation. Okay, so here complement, in the complement fixation test, here the serum with antibodies with serum is added, and then antigens are bind with them, and here also a control is present, control tube is also present where no antibodies is added. Okay, and after the addition of antigen, so antigen antibody will be bind to each other and then hemolysin sensitized red blood cells serve as an indicator. So red blood cells are added which act as a indicator. So RBCs or red blood cells settle into a pellet. So no lysis here. So this is reactive. Okay. So in this way complement fixation test is done. Sorry. So move on to the next question, that is Paul-Bernal test is a passive agglutination test or hemagglutination test or coagulation test or precipitation in gel test. So Paul-Bernal test is a B, that is hemagglutination test. Okay. So the next one is Lansfield's grouping of streptococci is a slide flocculation test or tube flocculation test or tube agglutination test or ring precipitation test, and the right answer is option D, that is ring precipitation test. And landslide grouping of streptococci is a ring precipitation test. So what is ring precipitation test? So this test is carried out by layering the antigen on the immune serum. So here at first the antibodies are added and then the antigens are added. So in the middle a ring will form, precipitin ring will form like this, a precipitin ring form. So in this way antigen antibody complex can be seen. Okay, and here the Lansfield grouping, this Lansfield grouping is a system of classification that classify catalase negative gram positive cocci based on the carbohydrate composition of bacterial antigens found on their cell wall. So basically for serological test Lansfield classification is done for streptococci. So by this Lansfield classification, streptococci are classified into these groups. Okay, so streptococci classified into many groups from A to K and one or more species per group, and classification based on the carbohydrate carbohydrate antigen present on the cell wall of them. Okay, so this is the Lansfield grouping of streptococci. So the next question is endotoxin is heat stable, antigenic, can be toxoid, or all of the above, and the right answer is option A, that is heat stable. So endotoxin is a heat stable. So endotoxin basically present in the outer membrane of the gram negative bacteria, that is lipopolysaccharide, which are heat stable. Okay, so the last question is C5 and C8 are synthesized in spleen, liver, macrophages or intestinal epithelium, and the right answer is option A, that is spleen. So C5 and C8, which are the complement protein, are synthesized in spleen. Okay, so these are the all in this series in this video. Okay, and thank you for watching this video, and we will see you on our next video, that is part 11 immunological MCQ. Okay, thank you for watching this video.